This is part one on a lecture on the genetic regulation of longevity in Drosophila. And one kind of thing that I wanted to start by talking about in terms of genetic regulation, and when we think about Drosophila as a genetic model organism versus yeast or worms, is that Drosophila is actually considered rather genetically complex compared to those other two organisms. In yeast and in worms, there's sort of a one-to-one gene to protein to phenotype relationship, where one gene is responsible for making one type of protein, and that protein exerts one specific phenotype. But genes in Drosophila are relatively pleiotropic, or can have multiple phenotypes. And that makes studying aging in them a little bit more accurate, but also more complicated, because it becomes difficult to track the genetic origin of different phenotypes, especially those associated with aging and longevity. And so, taking that into account, we're going to talk a little bit about what we do know about the genetics of longevity and aging from studying Drosophila. We're going to talk about the Drosophila life cycle, as well as what diapause is and how it might affect lifespan. And then we're going to talk about several different pathways involved in lifespan extension and longevity. And so to start off, this is the life cycle of Drosophila melanogaster, or the common fruit fly, illustrated here on the right. And Drosophila go through 6 stages from embryo to adulthood. They go through 3 larval stages, the first, second, and third instar larvae. And then they become or enter the pupal stage, and then ultimately become adults. And you can see the timing for that laid out here on the inside of this diagram. It takes about 9 days for an embryo to become a fully grown adult. And females, at least, become reproductive almost immediately after reaching adulthood, maybe within 12 hours. And they remain reproductive for 15 to 20 days after that kind of beginning of adulthood point. A fly can live up to 50 days, and so anytime between sort of this 9-day point and this 50-day point, we can study aging, right? Because after the flies reach reproductive adulthood, any point after that would be considered no longer development, but aging. And what's nice about flies is there's actually a pretty extended reproductive period as well as post-reproductive period, so we can start studying aging in both of those time periods as well. And so that is sort of the development under normal conditions, but under stressful conditions, like many other insects and invertebrates, Drosophila can enter what's known as a diapause, or a period of suspended development, or sort of suspended animation, where they are no longer developing reproductively. And diapauses usually occur during unfavorable or stressful environmental conditions. And so this image here, you can see that at the optimal temperature, this fly signals for ovarian growth and ultimately reproduction. But at an unoptimal or an unfavorable environmental condition, like low temperature, these flies will sense that environment and respond accordingly by entering diapause. And the main hallmark of a diapause in Drosophila is that they are not reproducing or producing any eggs. And that's due to a decrease in the ovarian growth from the beginning. And so Drosophila diapause is hallmarked by the fact that there is no reproduction. The flies will also decrease their metabolism and increase their stress resistance, usually through increased expression of stress resistance genes, or sort of anti-stress genes. And all of these things together will help Drosophila survive the sort of unfavorable environmental condition long enough to ultimately come out of diapause and then begin reproducing when things get better in the environment. And from studying diapause, and the fact that there are sort of these increased genes associated with anti-stress expressed during diapause, researchers looked at these anti-stress genes and also sort of implicated them in longevity or extended lifespan in flies as well. So two anti-stress genes are highlighted here from this table 5.3 in your book. The first of which is an SOD, or superoxide dismutase, and SOD is basically prevents against the formation of free radicals, and so its overexpression can actually help to extend longevity. Heat shock protein 70, or HSP70, is a heat shock protein, which is a type of molecular chaperone involved in helping to fold, prevent misfolding, and repair proteins. And in addition, when you have an overexpression or a lot of HSP70, you can increase longevity as well. And this is because both of these genes are involved in sort of that anti-stress response. And by having more of these stress response genes around, can actually continue to resist some of these stresses long term, and that can lead to increased lifespan or longevity. And one of the most well-studied anti-stress genes that extends lifespan in flies is the Methusalah gene, or MTH gene. And Methusalah encodes a protein called a G protein coupled receptor, or a GPCR. It's actually expressed in neurons. But beyond that, the role of this GPCR and the specific mechanism for how it can extend lifespan, i.e. through which signaling pathways it works, is relatively unknown. What is known about Methusalah gene, as well as the protein, is that flies that have extra copies of Methusalah, or transgenic flies, actually have a very extended lifespan, as you can see here in the survival curve on the right. With wild type in blue and Methusalah life curve in red, you can see that Methusalah extends lifespan. And in addition to extending lifespan, Methusalah also has a very, very strong resistance to stress. And so down here, you can see some different types of stress and the average survival for flies when these different stresses are applied. Paraquat is an oxidative stress. It generates free radicals. It's just a kind of compound that you can add to the media, and it will induce oxidative stress. Starvation is another type of stress, and then high temperature, or temperature at 36 degrees, which for flies would be extremely stressful because I believe they normally live around 25 degrees. And so in all of these cases, in both males and female flies, transgenic Methusalah flies, or flies that have extra copies of the Methusalah gene, have increased survival in response to all of these different stresses, right? So you can see that illustrated really well here with the oxidative stress from paraquat. Those Methusalah flies survive almost twice as long. For females, the Methusalah transgenic flies survive 6 times as long, almost, for males. And that increase in survival is seen under starvation conditions or starvation stress, as well as high temperature. And so there is some link between this GPCR, Methusalah, and extension of lifespan, as well as an increased response to stress when there's a lot of copies of Methusalah around. But the total mechanism, as I said before, is not really well understood.